Whether God created mankind or we just evolved has been a contentious issue within Christianity. The human and chimpanzee genome projects were supposed to definitively answer the question, but they didn't. However, recent gene expression studies now shed light on the issue. The number of genes in the genome of a human being is only about 300 genes more than that of a roundworm. So do 300 genes turn a 1,000 cell roundworm into a 40 trillion cell human being? Whole genome sequencing has shown that the human and chimpanzee coding genes are 98.7% identical. So can a 1.3% difference in gene sequence turn this into this? These two butterflies look different, but both are genetically identical, with the differences due to temperature during development. Sexual determination in alligators is not genetic. Instead, alligator eggs that develop at a temperature of 86 degrees and below all develop into females. Eggs that develop at temperatures of 93 degrees and above all become males. So male and female alligators are genetically identical. In the human body, there are over 200 different cell types that are quite different, but all are genetically identical. So it's clear from these examples that the genotype, or the genetic sequence of bases, is not the only factor that determines the phenotype or appearance. The phenotype is often controlled by the amount of gene products that are expressed. ENCODE was launched in 2003 to identify all functional elements in the human genome. This massive 10-year project generated more than 15 terabytes of data. Even though only 2% of the human genome codes for proteins, the ENCODE data shows that 80% of the genome was functionally involved in the production of those proteins. At least 75% of all the DNA in our genome is being transcribed into RNA. Is it just bad design, or is it actually doing something? In nearly all genes that contain multiple exons or coding regions, the pre-mRNA is spliced in alternative ways, so multiple protein variants can be produced from the same gene. This splicing is controlled by the non-coding introns to regulate gene expression. This is a principal components analysis of alternative transcripts in 6 ape species, showing large differences between us and them in the human brain. In the heart, we find less variance, as expected. Scientists have examined total gene expression in human versus ape-induced pluripotent stem cells, which are a proxy for embryonic stem cells. In comparing humans to chimpanzees, we find that over 10% of the genes are overexpressed in humans and another 10% overexpressed in chimpanzees. We find similar results comparing humans to bonobos. However, comparing chimpanzees to bonobos shows virtually no differences in gene expression. So even though human and chimpanzee coding genes are only 1% different, the expression of those genes differ by about 20% in the developing embryo. MicroRNAs are small RNA molecules, usually about 22 base pairs in length, that control the expression of about half the genes in the human genome. MicroRNAs are made in the nucleus and then transported to the cytoplasm, where they are extensively processed. The mature microRNA binds to a specific sequence found in the mRNA of its target gene, blocking protein translation and hastening mRNA degradation. Even though humans have genes that are nearly identical with other mammals, their microRNAs are not. 29% of the microRNAs are specific for hominoids. Another 26 are common between hominoids and monkeys. Less than a third are in common with other mammals, and only 16% in common with non-mammalian vertebrates. So our microRNAs are quite different. Although long non-coding RNAs are a relatively recent discovery, they have powerful regulatory effects on the genes they control. Long non-coding RNAs can bind mRNA like microRNAs, blocking translation. They can also bind proteins, regulating their function. Or they can bind microRNAs, preventing their inhibitory function. Some long non-coding RNAs recruit transcription factors to regulate gene transcription. Still other long non-coding RNAs bind to mRNA, causing it to be alternatively spliced or degraded. So, long non-coding RNAs can regulate gene expression in numerous ways. This is a table of a comparison of long non-coding RNAs to mRNAs for humans versus other mammals. The first column shows that mRNA sequences of humans match pretty well with most mammalian species, even cows and mice. However, the second column shows that long non-coding RNAs from humans don't match well with most other mammals, and in fact are pretty close to what would be expected in a random sequence. This is a heat map of long non-coding RNA expression compared to humans, to rhesus macaques, cows, mice, and rats for different organs. Differentially expressed link RNAs are found mostly in human brains and testes, so link RNAs are an important part of human uniqueness. For many years, pseudogenes were evolutionists' favorite apologetic for the veracity of neo-Darwinian evolution, since they were claimed to be merely evolutionary relics of duplicated genes. However, recent studies show that many pseudogenes are involved in gene regulation. The PTEN gene codes for a phosphatase protein that regulates cell division to prevent cells from dividing too rapidly. Mutations of the PTEN gene are responsible for the development of some cancers. The expression of the PTEN gene is controlled by a microRNA that binds to the mRNA, preventing it from being translated. However, the pseudogene of PTEN produces a similar transcript that binds the inhibitory microRNA, allowing the tumor suppressor protein to be translated. In this way, the PTEN pseudogene prevents cancer. Histones are proteins around which DNA is wrapped. 56% of all genome space is subject to histone modification, which regulates gene expression. The fundamental organization of the eukaryotic DNA is the nucleosome. It consists of a core of 4 pairs of histone proteins around which the DNA is wrapped, with histone H1 on one end. This is a representation of how the nucleosomes are organized together, making this flower-like arrangement. These flowers are stacked on top of each other to form long threads. The threads are coiled, and those coils are further coiled in what has been called supercoiling, producing the chromosome. This is how 2 meters of DNA can be packed into each cell. Each of the 4 core histone proteins has a tail domain that is modified to control gene expression. Each of these tails of these histone proteins can be modified in 4 different ways. There's acetylation, methylation, phosphorylation, and ubiquitination, which control gene expression. This complex regulatory mechanism has been called the histone code. Scientists have compared histone modifications in prefrontal neurons for humans and chimpanzees, showing that numerous loci on each chromosome are enriched in humans or enriched in chimpanzees. 15% of human DNA is in open chromatin regions, which are actively involved in transcription. Open chromatin are regions in DNA where histones are absent or repositioned. Because these areas are bare DNA, they are more sensitive to the DNA digesting enzyme DNase I, which is how they are detected. These sites include enhancer and promoter regions, allowing access to transcription factors to enhance gene transcription. This is a comparison of locations of these DNase hypersensitive sites, comparing humans to chimpanzees. Most of these differences are not found in promoter, exonic, or untranslated regions, but are found in the intronic and intergenic regions where they control RNA splicing and transcription. 8.5% of human DNA consists of short sequences that bind transcription factors which regulate gene expression, usually by increasing the rate of transcription for the particular gene with which they are associated. Although we think of genes as being linear, with a transcriptional start site, a promoter, and enhancer elements, the reality is that DNA is not flat, but bends and curves. So enhancer elements can bind transcription factors, promoting the binding of RNA polymerase complexes to enhance gene transcription. This is a comparison of CTCF transcription factor binding sites in humans versus gorillas for iPSCs, showing that over half of these sites are unique to each species, even though our coding sequences are nearly identical. CpG sites account for 4.6% of the human genome. Methylation of some of these CpG sites downregulates gene expression. Genome-wide methylation and gene expression shows that the vast number of CpG methylation sites are differentially methylated between humans and chimps. So, we have seen that gene sequence alone does not account for human uniqueness. The reason why you are not a chimpanzee are due to the design of multiple redundant molecular components that control gene expression. Not everybody was happy with ENCODE's proclamation of 80% functionality. Dan Grar, in a commentary, said, "Only sequences that can be shown to be under selection can be claimed with any degree of confidence to be functional." And he subtitled his article, "The Evolution-Free Gospel of ENCODE." So how much of the DNA is subject to selection? Numerous studies, published since 2003, have estimated that only 6 to 8% of the human genome has been subject to natural selection. Since 80% of the human genome has some function, but less than 10% of the human genome has been subject to natural selection, it would seem that evolution through natural selection does not explain 70% of genome function. So if natural selection is the driving force for evolution, most of the human genome has not evolved, but is designed. This article in Nature came out last October, asking if standard evolutionary theory needed to be modified. Some evolutionary biologists were pushing for what has been called an extended evolutionary synthesis. Standard evolutionary theory has become a theory of genes, where random genetic mutation is acted on by natural selection, resulting in the eventual fixation of advantageous genes into the genomes of populations of species. The extended evolutionary synthesis is a new theory of forms, where environmental influences, developmental plasticity, and developmental bias lead to phenotypic changes in species in the absence of genetic changes. Eventually, those phenotypic changes get hardwired into the genetic sequences of populations. The problem with such a theory is that there's no data supporting the idea that such a mechanism actually exists. Thank you for watching. May God bless you.